Hello, my name is Dr. MarjaneLeBagousse. I'm the medical director of HalioDx, a French-based company in vitro diagnostic for precision oncology. That's why in my background, I'm saying hello from a sunny day in Paris to all of you. And on behalf of all our teams, I do thank AWC organizers for this Digital Health Congress to invite us for this talk about Immunoscore, which is our immunoassay, a tissue-based immunoassay done by digital pathology and using artificial intelligence to enhance the performance of this assay in oncology decision-making. We are a French company in south of France, in Marseille. We have our headquarters, and we have an international footprint as well in USA. In Richmond, we are present. Let's share my slides. So first, let's have an overview about Immunoscore and the concept of our immunodiagnostic test. It is about the host immune response against the tumor. The host immune cells are infiltrated in a solid tumor. So as you see in this picture, the green dots are immune cells. And specifically from the family of CD3 and CD8 lymphocytes, we measure this infiltration into the tumor and around its margin, which we name invasive margin. So this infiltration gives the score about the status of the tumor and its risk of relapse. So accordingly, it can guide a therapy prescription. We develop with the license of Immunoscore different strategies. First, guiding therapy prescription, but also we help to optimize immunotherapies development, collaborating with different companies and in pharma area. And we help to predict response in immunotherapies when it is about companion diagnostic testing. The concept can respond to all these strategies. And we perform Immunoscore testing in our centralized lab of HalioDx. We have two in the world, in Europe and in USA. We are an organization with 200 employees. We have our own premises for research and development and laboratories and manufacturing units. And we are fully certified, and we meet regulatory requirements, and we own expertise in that, with an international footprint, Europe and USA-wise. A strong scientific background endorses HalioDx and its works, with Jérôme Gallon himself as co-founder, who is the head of immunology research in the public research organization in France. And he has been co-founding this company after years and years of works on Immunoscore concept. We have strong academic collaborations as well, and we have key business relationship with industry with different references, as you see on my slide. In our company, we work in two main business units, business unit of pharma and business unit of diagnostic. In pharma, we work through different services and products like Immunogram, for example, which is a global group of relevant biomarkers that give a complete image of immune contexture of a tumor, with Immunoscore for clinical research or Immunoscore as a companion diagnostic development. And in diagnostic business unit, we deliver Immunoscore as a test. It is an available standardized test for oncologists, and it is indicated in colon cancers and in rectal cancers. It is evidenced by strong clinical research, and it has been already tested in clinical studies with 6,500 patients in focused and dedicated designed clinical studies. And we have a market access plan with private medical insurances reimbursement plan already available in some countries. And we have also commercial channels in worldwide markets. The roadmap of HalioDx Group is to develop Immunoscore as the standard in clinical management of colorectal cancers and to expand the utility of Immunoscore to different solid tumors. We have our pipeline of development. As we said, this approach, this assay is applicable potentially in several solid tumors. We also partner with pharma companies to identify immunotherapy responders, and all this based on the concept of each cancer patient needs an assessment of his immune response to the tumor. Let's speak about the ready-to-use and available test Immunoscore for precision oncology today. This technology goes to qualify and quantify the CD3, CD8 cells into the tumor through an immunohistochemistry stained slide of a tumor sample. It is a robust and standardized assay that goes to identify categories of Immunoscore, categories of infiltration of those immune cells into the tumor from 0 to 4. So we have 5 categories of Immunoscore and regrouped in 2 main risk groups. The higher the infiltration is, the higher the Immunoscore category is. The medical utility of Immunoscore is to be an adjunct to the TNM classification system, which is the gold standard for classifying tumors, and it has been working since decades ago, but it is lacking a refinement and precision. And with immune parameter, we bring precision to TNM classification in order to give a very precise understanding of the risk of relapse and the clinical outcome of our patient, and accordingly to decide about the treatment strategy. This is a diagram from the validation study of Immunoscore. It was a Cox multivariate model comparing different risk identifiers for a tumor. The first identifier, for example, the TNM staging, which is the gold standard, T for tumor size, N for lymph node infiltration, and M for metastasis. And we put also differentiation of the tumor, the VLP, MSI status, and sidedness of the tumor, and a setting of risk stratifiers. And when we add Immunoscore to these risk stratifiers, we see as an independent biomarker, Immunoscore stays as the strongest one in the contributive role to identify the clinical outcome and the survival chance. This is for the 5-year survival chance, and the place of Immunoscore in this diagram is the strongest one. Almost half of the judgment about the clinical outcome is brought by this independent biomarker. So commercially speaking, we are rolling out our presence in the USA and in Europe by a direct dedicated staff. And we have a network built up in different countries. And in Korea, we are looking forward to partnering with a dedicated representative. And we have a value-based pricing implementation in different markets. Let's speak about the area where Immunoscore is applied in colorectal cancers, in colon settings, actually. We know that today colon cancers is still a very vast area of cancers where we have this validation of Immunoscore. We have already 2 million of people every year being diagnosed with colon cancers, unfortunately. And 1 million of these patients are dying every year, despite all the diagnostic and the clinical management regimen and different therapies strategies which are existing. But still, we observe caveats in this clinical management, and we observe that for stage 2, for example, we have still 15% of relapse in these tumors, despite of a very good clinical management with available practice. And in stage 3, we have 60% of overtreatment with chemotherapy. So we can figure out immediately how much side effects and prejudice to quality of life we are bringing to this area. And we need precision, we need accuracy for this management in these settings. And this is responded by Immunoscore as a biomarker in precision oncology. An extensive clinical validation sets of publication does exist, with the first study over 3,500 patients, which was composed of stage 1 to stage 3, specifically colon cancers. Then for stage 2, we had a dedicated cohort of more than 1,000 patients in which we were able to demonstrate the de-escalation capacity of Immunoscore for chemotherapy. And for stage 3, the ability to predict the benefit of chemotherapy in patients who are patients taking benefit from their chemotherapy in stage 3 has been already validated in 2 different clinical studies published with more than 2,000 patients. For the future, we have ongoing planned studies with more than 2,000 patients in our cohorts for clinical impact and revalidation of predictive ability. Some figures and some facts. After 15 years of research, this test has been commercialized, so a very strong endorsement with data and translational and proof of concept research behind this, over thousands and thousands of patients, and now a presence in 25 countries in the world with reimbursement in private sector. And for public sector, national submissions are ongoing. Our assay is CLIA lab certified. For Europe markets, it is CE-IVD marked, and it is a performance test which uses digital pathology with artificial intelligence based on a very strong and specific clinical algorithm. Actually, this algorithm is proprietary for HalioDx, and it is made over a very strongly designed and important clinical study over thousands and thousands of cases. And this is quite precise for making decision now based on the report of Immunoscore. A slide of Immunoscore could contain 80,000 of T cells read out with a machine analyzer. So the accuracy, reliability, and precision is brought by this test. Immunoscore was validated by the publication of a consortium decision in 2018 in the Lancet journal. It was a consortium of 13 countries from the Society of Immunotherapy in Cancers. It was an international study strongly designed and published in this journal. So clinically validated in localized colon cancers. The test was available for oncologists in 2018 and 2019. 1 year after that, WHO published its latest series of blue books with recommendations for pathologists in the world. And in this reference book, WHO included host immune response as a desirable and essential criteria in a colorectal cancer report, pathology report. So this is recommended for pathologists, and behind this recommendation, the reference stated by WHO is the reference of Immunoscore, because actually Immunoscore is the unique available test to assess the immune response at the tumor site. After pathologists, there was the European Society of Medical Oncology, the worldwide medical oncology guideline edition of their localized colon cancers embedded Immunoscore in their guidelines, and they recommend to use this test for decision-making in stage 2 and stage 3 colon cancers. So pathologists and oncologists have now their references and their recommendations. The report is given in this type, so with categories of Immunoscore, with the groups of risk. And the higher this infiltration in the tumor is, the higher the Immunoscore is, and the risk of relapse in these good responders is low. So a high Immunoscore is a low-risk tumor. A low Immunoscore is a high-risk tumor, and accordingly, we take the clinical decision. It is to be kept in mind that this test is completely non-invasive. It is done on available resected tumor. After the surgery, resection of the tumor, we send a sample to the central labs of HalioDx. The test is made and the report comes back to the clinician. No specific preparation procedure is needed, neither in pathology or in surgery. The sample is available and sent to the central labs, and the report is get back. The workflow is quite easy. The sample is stained in our labs. It is digitalized and it is analyzed by image analyzer, and the report is produced, and the whole turnaround time is maximum 10 days, and the shipment is taken in charge by HalioDx. Here, this is the process. I go quickly. Here we have the image of the tumor sample sent to our lab. For example, here we see the segments of the sample with the segment of the tumor tissue in red, the segment of non-tumor tissue or healthy tissue in blue, and in green we have the epithelial tissue. So this image is digitalized and it is uploaded in our image analyzer, and then an automated classification is made by the machine, and a manual adjustment by a trained user, an individual, is made in order to improve the classification of this automated process. And then at this step, an automated invasive margin is defined, and then the report is produced and controlled by an expert. So the intervention of an individual to check and to improve the classification of this segmentation is key and is important. Now let's speak about artificial intelligence with deep learning that we use to improve the training of our machine for image analysis and to improve that step which was controlled by human interference. To train our image analyzer, first we upload images by themselves, scanned images, and with annotations. This is the most important part of images nurtured, let's say, in our machine. Then in a second step, we nurture the machine with different models, and then the machine is able to segmentate the image. But to improve the process, we also nurture the machine again here at this step with testing images. A certain percentage of slides are uploaded here to control, and in this way we are able to get very precise segmented images from our machine. The architecture of neural network used in our analyzer is the so-called UNet architecture, which is quite known in medicine for the most precise segmentation of images. This goes likewise in brain. The input image data is given into the machine. It goes to expand, and then the information is again compressed and come out as output in a segmented map. So this precision and the function of this system, the neural system, is quite precise, and it gives a very accurate image output data. The metrics we use in our machine is the metrics of accuracy and intersection over union. The accuracy being the number of predictions from all the predictions made, of course, and intersection over union is calculated by a report of area of overlap. As you see here, the ground truth and the image should overlap, and calculated, reported to the rate of area of union, which should be ideally 1. So this report is computed for all tissue classes. Also, it is average for all tissue classes, and then it is computed also for tissue only. So these metrics are used for our machine, and we nurture the machine with different settings of image. As we said, setting for training the machine, then for validating, 10% is kept for training the machine, for validating the training, and 10% for testing that, for controlling. The parameters used in this are the level of the image, but also the context level, which is the level made in different context of pixels with different tunings. And then the border probability is a parameter important as well. In the borders of the sample, the machine should be able to attribute the right segment to the right class of the tissue. That's why we also nurture the machine with class weights. So class of epithelium, class of tumor tissue, or class of healthy tissue. And these are different models we use to know which type of model is the most performant in terms of accuracy or intersection of union. Here with accuracy, you see that we have used different models, and the blue model is the most performant one. As you see, in terms of probability for accuracy, we are in a very good situation. And we have used for this model one level image and 2 context level image, and we have activated the capacity of the machine to distinguish the border probability. So activating this capacity with these levels and context levels has given the best model to our machine. For intersection of union, the same type of exercise has been made, and again, the blue model is the best performant one. As you see, the result is very precise and very satisfying. In the left side, you have an expert annotated sample, and in the right side, you have machine annotated sample, which is quite precise, and you have parameters of accuracy and intersection of union on my slide, as you see. And you have, again, other examples with a very satisfying precision for every class of tissue. And in this overview, you see the workflow, which is composed of some steps, with first importation of data of image and slide to the machine, then automated image processing with detection of positive immune cells and segmentation of the image. And the image is registered, and then a manual review is made to define the invasive margin. And then a validation is made by an expert to produce the report. And the expert review controls all this to have a very precise report coming back to the clinician. So the tissue classification is a key performance parameter, as we see, and we improve it by manual adjustment, but this improvement can be made by artificial intelligence. That is what we do with deep learning into our machine, and we will have a streamlined digital pathology workflow. Thank you very much for your attention. If you need any supportive data or any contact for the Asian market, our responsible is Bertrand Farneau. And for any need for medical or scientific information, I'm at your service and very happy to answer to your questions. Thank you very much and have a good Congress.